right, let's talk about cell growth and division. So how cells get a little bit bigger and how they make copies of themselves. So we're going to talk about stages of the cell cycle, how that cycle is regulated, how it's controlled. We're going to talk about why it's so important to have this control over the cell cycle, and then we'll talk a little bit about the different stages of the cell cycle, which are mitosis and cytokinesis. So we have two different types of general types of cells in our body, somatic and germ cells. So somatic cells are most of your body cells. Germ cells are also known as gametes. These are egg and sperm cells. So these are very, very uncommon in comparison to somatic cells, because if a cell is not one of these gametes, it is not a sperm or egg cell, it is a somatic cell. So all of the other cell types in your body are somatic cells. These are going to have a homologous pair of chromosomes, which means that there are 2 copies of each chromosome. Humans have 23 different chromosomes, and so when we have those homologous pairs, that gives us 46 chromosomes total. Having these 23 pairs, so 2 copies of each chromosome, makes us a diploid organism. Germ cells, or gametes, these are going to have only 1 copy of each chromosome. So they are not going to have 46 chromosomes in them. They are only going to have 23 chromosomes per cell, which makes them a haploid cell rather than a diploid. So the cell cycle, by definition, is a series of events in the life of a cell from the moment it is created until the moment it either divides itself or ends, when it has basically outlived its usefulness. There are 2 main phases of the cell cycle, which are interphase and mitosis. So interphase is where cells spend the vast majority of their time. This is when the cell is not actively dividing and making a new copy of itself. Interphase is further broken down into 3 subphases, which are the G1 phase, S phase, and G2 phase. So G1, also known as the gap 1 phase, or sometimes it's called the growth phase or first growth phase. So this is where the cell is starting to prepare itself for the process of creating another copy. So we need to do a little bit of growth and kind of cell maintenance in this phase. This is followed by the S phase or synthesis phase. This is when the cell is replicating its DNA so that the extra copy of the cell is going to have all of the chromosomes it needs. And then we have the second gap phase or second growth phase. This is continued cell growth and preparations for mitosis. So we are making sure that there are enough organelles to be split up between the 2 daughter cells. We're making sure that there's enough cytosol to go between the 2 cells, all of those structural support filaments. We need to make sure there is enough of all of these different essential parts of the cell to go into 2 separate cells. There is also a G0 phase. This is a resting phase that would come after the G2 phase in some cases. This phase does not always happen, where the cell finishes all of these growth and synthesis phases and then pauses for a little bit before starting mitosis. But this does not always happen. Very frequently, cells will go straight from the G2 phase directly into mitosis. But sometimes they do need to take a little bit of a pause before entering that mitosis phase. So this is a graphical representation of the cell cycle, and specifically mostly interphase. So mitosis is this little orangish colored pie piece here that has an M. That's mitosis, and everything else here is interphase. So our growth 1, synthesis, and growth 2 phases, as you can see, make up the vast majority of the cell cycle. So let's talk a little bit more about chromosomes. So again, we are replicating our DNA during that synthesis phase, giving each cell 2 copies of all 23 chromosomes. Each copy is referred to as a sister chromatid, so it is an identical copy. Sister chromatids are held together by centromeres, so we need to be able to link those together to keep them together initially until we are ready to split them into the 2 separate cells. So we talked a little bit about homologous pairs of chromosomes. So the homologous chromosomes would be represented by each of the colors here. So the 2 red ones here are going to be those homologous chromosomes, and then sister chromatids are going to be identical copies. So homologous chromosomes are not necessarily going to be identical. You get 1 of each from each of your parents. So that's why they're not going to be identical necessarily. They're going to contain the same genes on them. They're going to be the exact same chromosome, but the exact sequence of those nucleotides, the A, T, Gs, and Cs, is not going to be identical between the 2 of them because your parents are not identical. Sister chromatids are going to be identical copies of that chromosome because we replicated that specific chromosome, and ideally there are no mutations that are coming from that. Mutations do happen, we'll talk more about those later, but they do occur sometimes and make them slightly less identical, but cells, human cells at least, are generally very good at fixing any mistakes that happen during DNA replication. So cell division, or mitosis, is the process of actively splitting that cell into 2 identical copies. So we've produced a bunch of extra organelles and cytosol and our copies of our chromosomes. Now we are going to divvy up all of those contents to either end of the cell and then split that cell into 2 final copies. So mitosis is broken up into 5 phases followed by cytokinesis. So our 5 phases of mitosis are prophase, prometaphase, metaphase, anaphase, and telophase or teleophase, depending on how you want to say that. And each of these is really essential. Prophase is when those chromosomes form. So before prophase, the DNA is still in that chromatin form where it's kind of loose and uncoiled. Prophase is when we actually coil that into chromosomes. And these actually do become visible in the cell. And so you can kind of see down here in these microscopy images with that fluorescence microscopy that I mentioned before. We have the chromosomes highlighted here in green. All right. And so we start seeing some green forming here in prophase. And prometaphase, those chromosomes are going to start lining up along the center of that cell. And then we're going to have microtubules start to attach to those centromeres and attach to centrosomes at the ends of the cells. Metaphase is this final stage of lining up those chromosomes. Each of the sister chromatids has been attached to one of those spindle fibers or microtubules that's coming from opposite ends or opposite poles of the cell. Anaphase is when we are going to start pulling those sister chromatids apart from each other. All right, and now they can be referred to as chromosomes again. So those spindle fibers are going to pull the sister chromatids apart towards opposite ends of the cell. And as you can see, other organelles are starting to move towards opposite ends of the cells in both metaphase and more distinctly in anaphase. At this point, between metaphase and anaphase, the nucleus does not exist in the cell. The envelope around the nucleus has dissolved, it's broken down, so that we can divide these chromosomes, all of that genetic information, up into the resulting cells. In telophase, That is when we start to reform that nuclear envelope. So it surrounds all of those chromosomes that have been pulled to opposite ends of the cell, and we are reforming that nuclear membrane and the nucleus itself. The spindle fibers are going to continue moving in opposite directions, further pulling the cells, two halves of the cells apart. And then in cytokinesis is when the membrane of the cell is actually going to start pinching off in the middle, and it's going to keep pinching in closer and closer together until it meets the other part of the membrane, in which case it will reseal itself into two individual daughter cells at the end. That's true for animal cells. Plant cells have a little bit more complicated of a process because they also have cell walls, but we're not going to get into too many details about plant cells because this is for humans in this class. All right, so let's talk about control of the cell cycle. So the cell cycle has a lot of different checkpoints. So this is kind of like DUI checkpoints, if you want to relate it to something in real life that some of you maybe know about. So this is stages in the cell cycle when there is a check to make sure that we have all of the necessary organelles, all of the necessary cytosol, we've replicated our DNA correctly and formed it into the sister chromatids and stuff. We want to make sure everything is going the way it's supposed to. And as long as everything checks out, then the cell cycle will continue. If it is not looking okay, that cell cycle will stop so that it can either be fixed or the cell is going to undergo apoptosis, that programmed cell death. So the main molecule responsible for this cell cycle control is called cyclin. There's multiple types of cyclins. One group is cyclin-dependent kinases, or CDK. This is a group of molecules that work together with the cyclins, so cyclin-dependent. So it depends on those cyclin molecules. So together, these molecules are going to determine what the cell is doing throughout the cell cycle. Does it get past that checkpoint or not? This is really, really essential because if we have something wrong with the DNA or other issues with the cell, then this can result in cancer if the cell is allowed to continue throughout that process and make more copies of itself. So cancer is a loss of control in the cell cycle. So cancerous cells do not get a signal to stop replicating, and they are not getting these checkpoints happening, which is why we get so many mistakes, why cancer cells look really, really weird. They do not look the same as normal body cells, which is why we're able to look at them under a microscope and identify them pretty easily. You do need to be trained to do that. That's what a lot of pathologists do, is look for cells that don't look the way they're supposed to, and determine, is this maybe cancer or is this definitely cancer? So this cell cycle regulation is super, super, super important to prevent cancer. Most cases of cancer are caught during these checkpoints, and it causes the cell to undergo apoptosis and degrade itself, and then that stops cancer from continuing. But I guarantee all of you have had cancer at some point or another. You just don't know it because your body has taken care of it. It has all of these mechanisms in place to prevent stuff like that from happening. Obviously, it is not a perfect system. Plenty of people do still end up with cancer as a disease that sticks around and continues replicating those cancerous cells, but the vast majority of cases are dealt with by the body's mechanisms. So This is where we're seeing a lot of those checkpoints happening. So in the G1 phase, we do have a checkpoint here at the end. We're utilizing a couple different cyclins and CDK molecules here during that growth 1 phase. We have another cyclin and CDK present here in the synthesis phase, but no checkpoint in the synthesis phase, as you can see here. But we do have another checkpoint in the G2 phase using some other cyclin and CDK molecules. And this is that final checkpoint before we enter mitosis, but we do have another checkpoint in mitosis, basically making sure that we have the appropriate chromosomes and the proper number of chromosomes that have been pulled to opposite ends of the cell and are going to be part of those new daughter cells, because we need to check that too. DNA could have replicated beautifully, but if we are not splitting up the chromosomes and sister chromatids correctly, we can also have issues there. All right, and that wraps up our discussion about the cell cycle, and we will finish up this week with differentiation of cells.